Imagine you're buying a house
Okay.
and you walk onto the property, you look up and you see a solar panel on the roof.
Right.
And then inside, there is a toaster in the kitchen.
Pretty standard so far.
Yeah, but here's the thing. For the next 270 years, your friends, literally every contractor on Earth, they all argue that these two objects are completely fundamentally unrelated.
Wow. 270 years.
Exactly. You put them in different manuals, you give them different names, and you treat them as totally separate phenomena.
Just completely missing the big picture.
Right, because what everyone missed is that there are these microscopic wires hidden inside the walls, physically connecting the two.
Ah, I see where you're going with this.
Yeah, so that level of prolonged monumental misunderstanding, that is exactly what happened when the father of modern science looked at the organism we are diving into today.
It really is wild when you put it in that perspective.
Welcome to another deep dive. Today we are looking at a biological paradox that is, honestly, it's going to completely change how you look at the natural world.
It really will.
We are talking about Lichenomphalia umbellifera, which is commonly known as the Lichen Agaric, or the green pea mushroom lichen.
It's a truly mind-bending subject. I mean, when you walk through a dense, wet forest or, over a freezing alpine tundra, you might think you were just looking at dirt and moss.
Right, just the usual forest floor stuff.
Exactly. But in reality, you are walking past a hidden world of intense symbiotic negotiations.
I love that phrasing, symbiotic negotiations.
Yeah, and our mission today is to explore how this seemingly ordinary tiny mushroom is actually a rare hybrid organism. It's a Basidiolichen.
Okay, let's unpack this. Because we're going to dissect its unique chemistry, its microscopic farming techniques, and its incredible ability to survive the most hostile elements on Earth.
It really is a survivor.
It is. But before we can understand the mechanics of how this organism thrives, we have to look at the history, right?
We do. The history is fascinating.
We have to look at how humans spent centuries fundamentally misunderstanding what it actually is.
Yeah, the taxonomic history of Lichenomphalia umbellifera is, well, it's a perfect reflection of how difficult it is to study things that blur the boundaries of life.
Blur the boundaries. I like that. So where do we start?
To really grasp the depth of the confusion, we have to travel back to 1753.
Okay, 1753.
We are looking at the work of Carl Linnaeus, who we often call the father of modern taxonomy.
The guy who basically named everything.
Exactly. In his seminal work, Species Plantarum, Linnaeus encountered this organism in the wild, and he made a massive double mistake.
So he essentially looked at the solar panel and the toaster and classified them as two entirely different kingdoms of life.
He did. When Linnaeus looked at the fruiting body, the tiny mushroom part, he named it Agaricus umbelliferus.
Agaricus umbelliferus, got it.
He believed it was a standard saprotrophic mushroom.
And for you listening, a saprotroph is it's an organism that gets its energy by simply decomposing dead organic matter, right?
Right.
Like a rotting log or dead leaves.
Right, just your standard decomposer. But he completely missed its connection to the green, granular vegetative mat it was growing straight out of.
He just didn't see the connection at all.
No, he looked at that green mat and classified it as an entirely different organism altogether. He called it an alga, naming it Botrydina.
To a modern observer, that seems like, well, it seems like a glaring oversight.
It does. It really does.
How could someone who dedicated their whole life to categorizing the natural world miss the fact that the mushroom was physically growing out of the green mat?
Well, it comes down to the severe limitations of observation in the 18th century. And that was coupled with a fundamental global misunderstanding of what lichens actually are.
Right, because they didn't really have the concept of a lichen yet, did they?
Exactly. You have to put yourself in Linnaeus's shoes. The dual nature of lichens, the fact that they are a symbiotic partnership between a fungus and an alga living together as one unit.
Right, two things living as one.
Yeah, that wasn't formally proposed until Simon Schwendener published his dual hypothesis in 1869.
Wait, 1869? That is more than a century after Linnaeus's initial classification.
Precisely. So to Linnaeus's naked eye, the structural divide between a fleshy, delicate mushroom and a crusty green granular mat on the ground was simply too vast to bridge.
They didn't look like they belonged to the same kingdom, let alone the same individual organism.
Exactly.
So because Linnaeus split the single organism into two different taxonomic files in 1753, the scientific community just ran with it.
They absolutely ran with it.
I imagine this caused absolute chaos for the botanists and mycologists who had to follow his lead.
Nomenclature chaos is the most accurate way to describe it, honestly.
I bet.
Because the mushroom and the green mat were treated as separate entities, they each went on their own winding taxonomic journey for over 2 centuries.
2 centuries of getting it wrong.
Right. The mushroom was bounced around various fungal genera as mycologists tried to figure out where it belonged based on its shape and gills.
So what did they call it during that time?
It was called Omphalia, then it was moved to Omphalina, and eventually some placed it in Geronima.
Okay, so that's the mushroom's journey. And meanwhile, the green mat, the thallus, is having its own completely separate identity crisis in the botanical records, right?
It was an entirely separate track of science. Botanists and lichenologists were studying the asexual lichen thallus independently of the mycologists studying the mushroom.
That is wild.
The green mat was given names like Botrydina vulgaris and Caricium viride,
Okay, so total parallel universes.
basically. In fact, Linnaeus' original name, Bisis botryoides, became the type species for the generic names Phytoconis and Botrydina. Both of those are now officially rejected names in the International Code of Botanical Nomenclature.
So you essentially had parallel sets of scientific literature, separate conferences, and separate field guides being written about the exact same organism for hundreds of years, completely unaware of each other.
Exactly.
If you are an ecologist trying to track the health of the green mat, your data is totally disconnected from the mycologist tracking the life cycle of the mushroom.
Yep, decades of research, all treating the two halves as unrelated phenomena.
So how did we finally manage to put the organism back together?
The modern resolution required technology that Linnaeus couldn't have even dreamed of. Molecular phylogenetics.
Ah, DNA testing.
Exactly. By comparing DNA sequences in the lab, scientists were finally able to unequivocally prove that the hyphae, the microscopic fungal threads forming the delicate mushroom, were genetically identical to the hyphae weaving through the green algal mat on the ground.
So they finally found the wires inside the walls.
They found the wires. They proved they were one and the same continuous network.
That must have been a huge moment in the mycology world.
Oh, massive. This undeniable genetic proof led to a major taxonomic revision in 2002, formally creating the new genus Lichenomphalia to house these unique organisms.
But the renaming process wasn't entirely clean, was it? I remember reading there is a bit of historical irony tangled up in its official name now.
Yeah, there is a quirk in the rules of botanical nomenclature at play here. When you untangle the historical records, it turns out the earliest legitimate description of this specific Basidiolichen was actually Agaricus pseudo-androsaceus.
That's quite a mouthful.
It is.
Yeah.
It was described by a botanist named Bulliard. However, that was superseded by a sanctioned later synonym, Agaricus ericitorum, described by Persoon in 1796.
Okay, so technically, by the strict rules of taxonomy, the legitimate name for this organism is Lichenomphalia ericitorum.
Technically, yes. But the epithet umbellifera, derived from Linnaeus' original mistake, is so deeply entrenched in historical literature, field guides, and digital databases that it remains the primary identifier used by most researchers today.
It's a fungible name that just refuses to fade away, despite the rules.
Exactly. It just stuck.
It is a brilliant reminder for you listening that scientific truth is rarely a straight line. It is messy, it is highly iterative, and our understanding of the biodiversity of this planet can be severely limited by what our eyes immediately perceive.
Very true.
We have to constantly be willing to revise our assumptions when new tools arrive.
And what's fascinating here is how the physical structure of this organism dictated its history.
Right. The way it looks caused the whole problem.
Exactly. Now that we have solved the historical mystery of its identity, we need to look closely at that physical structure to understand why it was so easily misunderstood and how it actually operates in the wild.
So if you are out hiking and trying to spot this, the visual profile of the fruiting body, the mushroom itself, is incredibly delicate.
Very delicate.
We are looking for a pale brown to yellowish mushroom. It is tiny. To put the measurements into perspective, the cap is only about 5 to 15 millimeters broad.
So small.
That is roughly the size of your pinky fingernail, maybe expanding to the size of a quarter at its absolute largest.
Right?
It has a grooved cap, and what mycologists call distant gills, meaning the gills under the cap are widely spaced apart, and they run down the stem.
Yep. running right down the stipe.
And that stem, or stipe, is only 10 to 25 millimeters long. It is no longer than a standard paperclip.
It's remarkably tiny, and if you were to take that tiny cap, place it on a piece of dark paper, and let it drop its spores, it would leave a pure white spore print.
A white spore print. Good to know for identification.
Definitely. And under a microscope, those spores are ellipsoid, meaning they are shaped like little ovals, and they measure about 7 to 10 by 4 to 7 micrometers.
That's incredibly small.
It is. And crucially for identification, they are inameloid.
Let's break down that term, inameloid. Why do mycologists care about that?
Well, mycologists often use chemical stains to identify spores. One of the most common is Meltzer's reagent, which is an iodine-based stain.
Like the iodine you'd use on a cut.
Similar concept, yeah. When a spore is amyloid, it means it contains starches that react with the iodine, turning the spore a deep blue or black under the microscope.
Okay.
Because the spores of Lichenomphalia umbellifera are inamyloid, they do not change color when exposed to this iodine stain.
So they just stay their normal color.
Exactly. It is a key structural clue about the chemical makeup of the spore walls, helping differentiate it from look-alike species.
But again, the tiny paperclip-sized mushroom is just the flashy, ephemeral part of the organism. The true, permanent vegetative state is what caused Linnaeus to invent a whole new alga in the first place.
The part he completely misread.
Right. You mentioned the Botrydina type thallus earlier. What does that actually look like if you are staring at it on the forest floor?
The Botrydina thallus is a cryptic, dark green, granular mat.
Like moss.
To the naked eye, it might just look like a green stain or a thin layer of unusual moss on the dirt. But under magnification, it consists of minute granules or globules that cover raw, peat, damp soil, or rotting conifer wood.
So it's not leafy at all.
No, unlike the classic leafy or crusty lichens you might picture growing flat against tree bark, this granular mat is considered a somewhat primitive architectural arrangement.
Interesting.
Each microscopic green globule is a core of single-celled algae tightly encased in a protective network of fungal hyphae.
Like a little protective bubble.
Exactly. These hyphae interconnect the granules across the soil to form a robust perennial network.
I was reading about the cellular anatomy of those fungal hyphae, and there's some really bizarre structural omissions for a mushroom of this type. It is missing biological machinery that you would normally expect to find.
The microscopic oddities are significant, for sure.
Yeah.
Most notably, the hyphae of Lichenomphalia umbellifera completely lack clamp connections.
Clamp connections are something you see heavily referenced in mycology. What exactly is the fungus missing by not having them?
To understand clamp connections, you have to understand the dekaryotic state of many fungi in the division Basidiomycota.
Okay, dikaryotic state.
Unlike human cells, which typically have one nucleus containing all our genetic material, these fungal cells often contain two separate, genetically distinct nuclei living side by side in the same cell compartment.
Two nuclei, one cell. Got it.
When that cell needs to divide and grow, it has to ensure that the new cell also gets exactly one copy of each distinct nucleus.
Right, you can't just mix them up.
Exactly. A clamp connection is a specialized hook-like bypass tube formed by the growing cell. It physically routes one of the dividing nuclei around the cell wall barrier, acting like a tiny biological traffic cop to ensure equal distribution and maintain that dikaryotic state.
So missing this hook-like bypass tube means Lichenomphalia umbellifera has to traffic its DNA using a completely different method during cellular division.
It suggests the organism has evolved a highly specialized, perhaps simplified, genetic and structural pathway to manage its cellular division without needing that physical bypass.
That's fascinating. So it just found a workaround.
Basically, yes. Furthermore, it also completely lacks hymeneal cystidia.
Okay, hymeneal what are those?
These are sterile, often enlarged cells found interspersed among the spore-producing cells in the gills of many other mushrooms.
What do they normally do?
They act almost like biological bumpers or space makers to keep the gills from sticking together.
Oh, like spacers on a tile floor.
Exactly. But Lichenomphalia umbellifera manages its delicate gill structure without them.
Wait, I have to stop and ask a fundamental question here.
Sure, go ahead.
If this organism is a lichen, meaning it is a symbiotic partnership with algae, why does it even have a gilled mushroom in the first place?
That's a great question.
When I think of lichens, I think of those crusty, grayish-green patches you see growing on gravestones or the side of a cliff. I don't think of a mushroom with a cap and a stem.
That is the perfect question because it highlights exactly why this organism is so special. You have just identified the Basidiolichen anomaly.
The Basidiolichen anomaly.
Right. When we use the word lichen, we are not
Ooh.
talking about a single evolutionary family tree. We are talking about a lifestyle strategy.
Oh, so it's more of an action than an identity.
Precisely. Different types of fungi have discovered the lichen strategy of farming algae entirely independently of one another. However, the vast majority, over 99%, of all known lichenized fungi belong to a phylum called Ascomycota.
And Ascomycota fungi reproduce very differently than the mushrooms we buy at the grocery store, right?
They do. Ascomycota are often called cup fungi. They produce their spores inside microscopic sac-like structures, often nestled inside little cup-shaped fruiting bodies.
So almost all the typical lichens you see on rocks and trees are essentially cup fungi that have taken up farming,
Exactly.
which makes our little green pea mushroom a massive statistical outlier.
Less than 1% of all lichenized fungi are Basidiolichens. That means fungi belonging to the phylum Basidiomycota, which includes the classic fleshy gilled mushrooms we are familiar with.
So it's a 1%er of the lichen world.
It really is. The existence of Lichenomphalia umbellifera proves that complex mushroom-forming lineages within the Agaricomycetes class have independently evolved the ability to domesticate algae.
It is an evolutionary marvel. Instead of relying solely on decomposing dead wood or leaves, it has created a self-sustaining nutritional mode by building a greenhouse for algae.
That's a perfect way to put it.
Here's where it gets really interesting, because the physical structure of that green mat and the tiny paperclip-sized mushroom is just the housing.
It's just the physical building.
Right, to understand how this organism thrives in some of the harshest, most nutrient-poor places on Earth, we have to look closely at the microscopic tenant living inside that housing.
The partner in the symbiosis.
Exactly. Who exactly is the partner?
The primary photobiont, which is the photosynthetic partner providing the energy, is a single-celled green alga known as Coccomixa subellipsoidea.
Coccomixa subellipsoidea.
Specifically, researchers often focus on a strain known as SAG2-16-13.
That sounds like a barcode.
It does, but it's crucial. This particular strain of algae is so incredibly resilient to freezing and desiccation that it has actually become a model organism for polar microbiology.
Really? A model organism?
Oh yeah. Scientists study it to understand how any cellular life can survive the extreme conditions of the polar regions.
So the fungus builds those little Botrydina granules specifically to house this extremely resilient polar algae. It sounds like a highly targeted architectural project.
It really is. The fungus completely encases the individual Coccomixa cells in a dense three-dimensional network of hyphae.
Like wrapping it up.
Exactly. The architecture is brilliant. It isn't just a blanket thrown over the algae. The fungal wrapping is designed to maximize the surface area of the algae exposed to sunlight, allowing for peak photosynthesis, while simultaneously buffering the delicate algal cells against the violent temperature swings and moisture fluctuations of the external environment.
It is the ultimate landlord-tenant agreement.
Completely.
The fungus builds a climate-controlled, secure apartment on the freezing tundra, and the algae pays rent. And the currency of that rent is a very specific type of sugar, right?
Yes. The algae harnesses solar energy through photosynthesis to fix carbon from the atmosphere. It then transfers a massive portion of this captured carbon to the fungal landlord in the form of a 5-carbon sugar alcohol called ribitol.
But this trade introduces a massive metabolic problem for the fungus.
It really does.
When I first looked at the biochemistry of this exchange, I assumed the fungus just ate the sugar and grew. But it's not that simple, is it? The rent money is actually somewhat toxic to the fungal machinery.
It is incredibly dangerous for the fungus if it isn't managed with absolute precision.
Wow, toxic rent.
Yeah. This brings us to a fascinating biochemical defense mechanism embedded deep within the symbiosis. The fungus, like many organisms, relies on a process called glycolysis to generate its own internal energy.
Glycolysis.
Right. Glycolysis is the metabolic pathway that breaks down glucose to extract energy for cellular metabolism. However, environmental sugar alcohols, specifically the ribitol being pumped out by the algae, can severely inhibit key glycolytic enzymes within the fungal cells.
So the food the algae provides is essentially the wrong type of fuel for the fungus. It's like pouring diesel into an unleaded engine. It's going to clog up the system.
That's a great analogy. It specifically targets and inhibits an essential enzyme called phosphoglucose isomerase, or PJ.
Okay, PJ.
If the PJ enzyme is inhibited, the fungal metabolism grinds to a halt. It would be a fatal flaw in the symbiosis if the fungus hadn't evolved a highly specific rapid response workaround.
How does the fungus prevent the ribitol from shutting down its internal engine?
To protect its PGI enzyme, the fungus expresses an enzyme of its own called PYP1. PYP1 is a polyol phosphatase.
Polyophosphatase. Okay.
Think of it as a molecular translation service at the border of the fungal cell. As soon as the ribitol crosses over from the algae into the fungal hyphae, PYP1 immediately goes to work, converting the ribitol into other, heavier sugar alcohols, primarily mannitol or arabidol.
It instantly changes the currency.
Exactly. These converted forms, mannitol and arabidol, are perfectly safe for the fungus.
So it just launders the money right at the door?
Yes. It can store them in large quantities without disrupting its internal energy production, and eventually metabolize them safely when it needs the energy to grow the mushroom or expand the mat.
That's incredible.
The fungus has evolved a molecular filter to instantly convert the rent money into a usable currency before it can poison the system.
And the physical integration between these two organisms goes even deeper than just a chemical exchange in the dirt. There is some cutting-edge research regarding where this algae actually lives that completely upends how we view the anatomy of this lichen.
This is one of the most profound new findings in Basidiolichens.
What did they find?
Well, for a long time, the foundational assumption was that there was a strict spatial separation between the two partners.
Okay.
We believe the algae lived exclusively down in the green vegetative mat on the ground, and the mushroom that popped up into the air was purely fungal tissue, built entirely from the energy provided by the mat below.
The assumption was that the mushroom was just a reproductive tower, completely devoid of algae.
But recent microscopic and genetic studies have revealed that the algae isn't restricted to the basal mat at all.
Wait, really? Where else is it?
Strains of Cocamexis subellipsoidea have actually been found living inside the stem, the stipe, of the mushroom itself.
Wow.
This means the symbiosis is far more intimately integrated into the vertical reproductive structures of the fungus than anyone previously imagined. The algae is traveling up into the air with the fungus.
It blurs the line of what constitutes an individual organism.
It really does.
The mushroom isn't just a fungal structure, it is a shared biological vessel.
Exactly, a shared vessel.
But this incredible sugar economy and cellular integration alone don't explain everything. Lichenomphalia umbellifera doesn't just live in mild, comfortable forests. It thrives in the brutal, freezing elements of the Arctic and on high alpine tundras where the wind howls and the sun beats down relentlessly.
Some of the harshest places on Earth.
Sugar won't stop you from freezing or drying out to a crisp. How does it survive the physical extremes?
To survive those environmental extremes, the lichen relies on a highly specialized chemical arsenal.
Chemical warfare.
Basically, yeah. The physical coming together of the fungus and the algae triggers the production of what we call lichen acids. These are unique secondary metabolites that are primarily synthesized through the acetylmalonate pathway, which is also commonly known as the polychide pathway.
Let's contextualize how massive this chemical production is. When we talk about secondary metabolites in plants, we are usually talking about trace amounts.
Right, very tiny amounts.
But in this lichen, these chemical compounds can make up anywhere from 0.1% to 10% of the organism's total dry weight.
Which is huge.
And in some extreme environmental cases, up to 30% of the lichen's weight is just these chemicals.
30%.
That is an enormous biological investment. What exactly are these chemicals doing to justify that energy cost?
The compounds are typically deposited as microscopic crystals on the exterior walls of the fungal hyphae. They aren't just metabolic waste products. They serve vital, life-saving ecological functions.
So they are tools.
Yes. The literature highlights several fascinating compounds found in these types of lichens, specifically acinic acid, atranorin, and umbelliferic acid.
I was reading about acinic acid, and its versatility is astounding. If you are devoting that much body weight to a single chemical, it must be doing some heavy lifting. What is the return on investment for acinic acid?
Acinic acid is a biological powerhouse. In the intense, unshielded solar radiation of high alpine or Arctic environments, it essentially acts as a highly effective, localized sunscreen.
Built-in sunscreen.
Exactly. It absorbs harmful UV radiation, protecting the delicate algal cells underneath from light-induced DNA damage. Furthermore, it serves as a potent anti-herbivore defense.
How does a microscopic crystal stop an insect from eating the lichen?
It acts as a feeding deterrent. Acinic acid has been shown to be toxic or highly distasteful to foraging insects, mites, and their larvae.
So they take one bite and say, nope.
Precisely. If an insect attempts to graze on the lichen mat, the umbelliferic acid disrupts their digestion, retarding their growth and significantly increasing mortality rates. It is literal chemical warfare against predators.
And what about umbelliferic acid? That one caught my eye when looking at how the lichen handles environmental stress, particularly drought.
Umbelliferic acid is deeply tied to the lichen's ability to endure severe dehydration. In laboratory tests assessing antioxidant capacity, scientists use something called a DPPH radical scavenging assay.
Let's break down that assay for you listening. DPPH sounds like heavy lab jargon. What exactly are the scientists doing in that test?
Think of the DPPH assay as a chemical obstacle course. DPPH is a dark purple chemical compound that contains stable free radicals.
Free radicals?
Yeah, molecules that are missing an electron and are eager to steal one from somewhere else. In a biological cell, free radicals are dangerous because they steal electrons from DNA or cell membranes, causing damage.
Right, they destabilize things.
Exactly. In the lab, scientists put the purple DPPH solution into a beaker and add the umbelliferic acid. If the umbelliferic acid is a good antioxidant, it will neutralize the free radicals by donating an electron, and the purple solution will visibly turn yellow.
So it's a visual test.
A very clear visual test. When they put umbelliferic acid through this test, it demonstrated a massive 68.14% inhibition. The solution turned rapidly yellow, proving it is an incredibly potent antioxidant.
To really understand why that antioxidant power matters in the wild, we have to talk about the acetone rinsing experiment. I found this absolutely mind-blowing.
It's such a clever experiment.
Researchers wanted to prove that these chemical crystals on the outside of the fungus were essential for survival, so they essentially gave the lichen a chemical bath.
A team of researchers designed a brilliant experiment. They took living lichens from the field and carefully rinsed them with acetone.
Just standard acetone.
Yep. Acetone is a solvent that is highly effective at dissolving and removing the crystalline secondary metabolites, like the arsenic and umbelliferic acids, from the surface of the hyphae.
Okay.
Crucially, the short acetone rinse leaves the underlying cellular structure of the fungus and the algae completely intact and alive.
It is the biological equivalent of stripping a knight of their armor. You leave the knight perfectly healthy, but you take away their outer defense.
Exactly.
What happens to the lichen when it faces a severe drought without its chemical armor?
The consequences are immediate and severe. In harsh environments, lichens frequently dry out completely, a state of severe desiccation.
Bone dry.
Right. When cells lose their water, the cellular machinery undergoes massive mechanical and chemical stress. This specific stress generates reactive oxygen species, or ROS.
I like to think of reactive oxygen species as molecular wrecking balls.
That is an excellent analogy. Wrecking balls, ROS, are highly unstable, oxygen-containing molecules.
Because they're missing an electron.
Yes. And because they are unstable, they swing wildly around the inside of the cell, tearing apart lipid cell membranes, denaturing vital proteins, and shredding DNA strands through a process called oxidative stress.
Oh, man. Just destroying everything inside.
Under normal, armored conditions, the secondary metabolites like umbelliferic acid act as the defense force, chemically scavenging and neutralizing these molecular wrecking balls before they can cause fatal damage to the lichen cells.
So what happened to the strip knights in the experiment?
The acetone rinsing experiment proved the necessity of the armor perfectly. When the researchers subjected both groups to severe drought, the unwashed control lichens relied on their chemical antioxidants to manage the stress.
And they were fine.
Yeah. When water was reintroduced, those normal lichens bounced right back, recovering their photosynthetic capacity almost immediately.
Wow.
However, the stripped lichens, the ones missing their secondary metabolites, suffered severe permanent cellular damage from the unchecked ROS. Their ability to recover photosynthesis was significantly impaired.
So the wrecking balls just took down the house.
Absolutely. It definitively proved that this unique chemistry isn't just a byproduct. It is an active, essential survival mechanism for enduring the planet's most hostile, dry barrens.
If you've ever tried to keep a garden alive in poor sandy soil, you know the absolute struggle for basic nutrients like nitrogen.
It's tough.
Now imagine you are this tiny fungus, you don't have fertilizer, you are living on dead moss in the freezing cold, but because you are equipped with a localized food source, the algae, and a protective suit of chemical armor, you are built to conquer environments where almost nothing else can survive.
It's the ultimate survivalist.
Which brings us to its ecological niche. We are looking at rotting conifer logs, damp soil, sphagnum bogs, and Arctic barrens. Lichenomphalia umbellifera is a pioneer of what ecologists call retrogressive landscapes.
The concept of ecosystem retrogression is a fascinating inversion of how we usually think about nature.
How so?
Usually we think of ecological succession as a forward march of progress. Bare rock breaks down into soil. The soil grows pioneer grasses. The grasses give way to larger shrubs. And eventually, after a few centuries, you get a lush, complex climax forest.
Right, nature healing and building.
But retrogression is what happens when a landscape ages over thousands of years without any major renewing disturbances.
So a landscape without a massive wildfire or a glacier scraping it clean basically just gets old and tired.
Precisely that. Over millennia of continuous rainfall and weathering, vital soluble nutrients, particularly nitrogen and phosphorus, are slowly leached out of the soil, washing away into the water table.
So the soil just loses all its value.
The soil fertility steadily plummets. As the soil degrades and becomes increasingly acidic, the large, complex vascular plants, the trees and the demanding shrubs, begin to starve. They cannot maintain their mass and they slowly die off.
That's kind of depressing.
It's a natural cycle. The landscape transitions backward, from a thriving forest into a nutrient-poor heath or a barren, acidic peat bog.
And this is where our little green pea mushroom lichen steps in. It is the ultimate homesteader.
Exactly.
Imagine someone going completely off the grid into the middle of nowhere. Because Lichenomphalia umbellifera brings its own food supply, the algae fixing carbon directly from the atmosphere, it simply doesn't care that the soil is dead.
It doesn't need to compete for nitrogen in the dirt the way a vascular plant does.
It's self-sufficient.
In these nutrient-starved, retrogressive ecosystems, the lichen goes from being a marginal background player to a dominant pioneer species.
It takes over.
It thrives on the raw, acidic peat and colonizes the moss-covered logs that nothing else can digest. In doing so, it serves a critical structural role for the entire ecosystem.
Structural role, like holding it together.
Yes, its dense, granular vegetative mats spread over the bare ground, binding the loose peat together and preventing erosion from wind and rain. It stabilizes the very surface of these harsh, dying landscapes, holding the ground together.
Its adaptability is so extreme that its evolutionary cousins are pushing the boundaries of life even further. I was looking at the recent literature, and there is a mention of a newly described relative, Lichenomphalia altoandina.
Oh, that one.
The habitat this thing survives in defies all logic.
The discovery of Lichenomphalia altoandina is astounding. It was found in the high Andes Mountains of Chile, at extreme altitudes exceeding 3,000 meters.
That is high.
But what makes it truly remarkable is its substrate. It isn't growing on peat or dirt or rotting wood. It has been found growing directly on top of salt crusts.
Growing on a salt crust should be a biological death sentence for a fungus. Anyone who has ever salted a slug knows what salt does to soft tissue. It violently pulls water out of cells.
The process is called osmotic shock.
Osmotic shock.
The extreme salinity outside the cell creates a gradient that forces water to rush out of the cell membrane, leading to complete osmotic collapse and death.
So how does it not just shrivel up?
But L. altoandina demonstrates the extreme, specialized adaptability of the Coccomixa fungal symbiotic blueprint. It is hypothesized that the fungal metabolites we discussed earlier, working in tandem with a thick, jelly-like mucilage produced by the algae, create a localized, microscopic barrier.
A jelly barrier.
Right. This barrier prevents the extreme salinity of the crust from drawing the water out of the lichen cells. They have essentially engineered a microscopic, hydrated oasis sitting directly on top of a lethal salt flat.
Which raises a massive logistical question. If they've engineered this oasis on a 3,000-meter salt flat or a remote Arctic bog, how does a tiny, delicate, paperclip-sized mushroom make it up there in the first place?
It's a great question.
It can't walk there. This brings us to the organism's global distribution. Because this incredible adaptability means this pioneer isn't just isolated to a few tough spots. It has built a global empire across the northern hemisphere. It travels via what ecologists call the circumpolar highway.
If we connect this to the bigger picture of global biodiversity, Lichenomphalia umbellifera presents a fascinating case study in phylogeography.
Phylogeography.
Phylogeography is the study of the historical and geological processes that are responsible for the contemporary geographic distributions of individuals.
Okay, so tracking how they spread.
Despite occupying a highly specialized, extreme niche, it is one of the most widely distributed mushrooms on planet Earth.
How exactly does a tiny mushroom in the Arctic spread so far and wide?
The primary mechanism is atmospheric wind currents, aided potentially by migratory animals brushing against the fruiting bodies.
Just blowing in the wind.
Exactly. The spores produced by the mushroom are incredibly light and resilient to cold. A pivotal 2012 genetic study analyzing the variation of Lichenomphalia species globally found something remarkable when they compared populations across the Northern Hemisphere.
What did they find?
They discovered no significant genetic differentiation between the populations living in the bogs of North America and those living on the tundras of Eurasia.
So a mushroom growing in a frozen bog in northern Canada is genetically nearly identical to one growing thousands of miles away on a tundra in Siberia.
It indicates a remarkably high level of intercontinental gene flow.
They're constantly mixing?
Spores are constantly being swept up into the powerful atmospheric currents that circulate continuously around the North Pole, the circumpolar highway.
Wow.
This continuous global mixing of genetics prevents the populations on different continents from isolating and evolving into separate endemic species.
Because they're always cross-pollinating, in a sense.
Right. It also allows the lichen to rapidly recolonize suitable habitats as the climate shifts, such as immediately following the retreat of massive glaciers at the end of ice ages. It is a highly mobile pioneer.
But the story changes entirely when you look away from the North Pole and focus on the southern hemisphere. Does the mixing ever stop?
The tropics act as a massive, almost impenetrable geographical barrier.
Because it's too hot.
Exactly. The same 2012 study looked at rare occurrences of Lichenomphalia in the subantarctic regions, remote islands like those south of New Zealand.
Okay.
Because the vast, hot expanse of the equatorial tropics is entirely unsuited for this polar organism, transequatorial dispersals, meaning spores successfully making the journey from the Arctic across the equator all the way to the Antarctic, are exceedingly rare events.
When a spore finally does make that impossible journey and lands on a subantarctic island, it is completely cut off from the main global population. It can't crossbreed with the Siberian or Canadian mushrooms anymore.
And what happens when a population is completely isolated in an extreme environment for thousands or millions of years without any new genetic material flowing in?
They evolve into something new.
Speciation. The 2012 study identified divergent cryptic lineages in the subantarctic. To the naked eye, these subantarctic lichens look morphologically identical to the northern Lichenomphalia umbellifera.
They look exactly the same.
They have the same tiny mushroom and the same green mat. But genetically, they have drifted far enough apart in their isolation to be considered completely separate phylogenetic species.
That's wild.
It is a live, observable demonstration of how geographical isolation drives the evolution of new species.
This brings up a philosophical question about evolution that I have to push back on.
Let's hear it.
When we talk about symbiosis, we often talk about evolution like it's a straight line or a deliberate, conscious choice. Does a fungus just decide one day to become a lichen? How does a traditional mushroom, equipped to decompose dead wood, suddenly start building greenhouses and farming algae?
This raises an important question about the mechanics of natural selection. Evolution is blind. It is driven entirely by random genetic mutation and relentless environmental pressure. An organism never decides to change.
Right, it's just surviving.
But what makes the Hygropheraceae family, the broader taxonomic family that contains Lichenomphalia, so incredibly important to science is that it acts as a live evolutionary laboratory.
A live laboratory, how so?
Within this single family, and even within closely related genera, we see both fully lichenized species that farm algae and strictly saprotrophic, non-lichenized species that still just decompose dead matter.
So you have cousin mushrooms, closely related on the genetic tree, where one makes its living decomposing dead wood and the other makes its living farming algae.
Yes, and modern phylogenetic mapping, tracing the DNA back through time, proves something astounding.
What's that?
The transition from a decomposing lifestyle to a symbiotic lichenized lifestyle didn't just happen once at the very base of the family tree. It has happened multiple times, entirely independently, across different branches within the group.
That implies the genetic machinery or the underlying biological potential to form these complex farming partnerships is somehow latent or easily triggered in these specific fungi.
It heavily suggests that under extreme environmental stress, when dead organic matter becomes critically scarce, such as in those dying retrogressive peatlands or on newly exposed sterile rock left behind by a melting glacier.
Basically when they're starving.
Right, the evolutionary pressure is immense. The environment heavily favors and selects for any random mutation that allows a starving fungus to capture and utilize an alternative carbon source, like a passing alga.
It just takes whatever works.
It is a real-time genetic look at how life transitions from a solitary struggle for survival to complex interspecies cooperation. Symbiosis, in this context, is a profound survival strategy born out of absolute environmental necessity.
So what does this all mean? We have covered a 270-year taxonomy battle, intricate sugar economies, chemical sunscreens, osmotic shock, and global spore highways.
We've covered a lot.
To bring it all back down to earth, why should you listening care about this one tiny mushroom? What is the application of this knowledge today?
Lichenomphalia umbellifera acts as a crucial sentinel species for the health of our planet
Sentinel species.
because it relies entirely on a very specific strain of Coca-mix algae to survive, and because its biological machinery is so finely tuned to thrive in pristine, nutrient-poor environments, it is incredibly sensitive to human-made atmospheric changes.
It feels the changes first.
It is particularly vulnerable to nitrogen pollution, such as agricultural fertilizer runoff or industrial emissions raining down, and to the rapid warming of the Arctic and alpine tundras.
There's a warning system.
By continuously tracking the health, distribution, and genetic diversity of this tiny mushroom, scientists possess a highly calibrated, living instrument to monitor the real-time impacts of climate change on the planet's most fragile, vital peatland ecosystems.
It is a microscopic canary in the coal mine for the global climate, and understanding how it works leaves us with a truly profound final thought.
Absolutely.
If a classic guild mushroom, the exact kind of structure we have universally assumed for centuries to be a standard, solitary decomposer of dead matter, can secretly be farming single-celled algae inside its own stem just to survive the freezing harshness of the Arctic, well, what else are we entirely missing?
It really makes you wonder.
What if other common fungi, the ones growing right now in our own backyards or local parks, are secretly harboring microscopic, unseen, symbiotic partners that we simply haven't looked closely enough to find yet?
It forces us to entirely reevaluate our perception of nature. Are the boundaries between individual organisms far blurrier than we ever imagined?
It might just be.
The natural world might be far less about isolated individuals competing for scraps, and much more about incredibly complex, invisible microscopic cooperations.
I think Linnaeus would be utterly stunned if he knew the truth about his broken toaster and solar panel. Thank you for joining us on this deep dive. The next time you are out on a hike and you see a mossy log or a patch of bare, peaty soil, take a second to look twice. You might just be looking at a 270-year-old paradox, quietly farming the sunlight.